Well, good morning, everyone, or good afternoon. Welcome back to the Cerebral Blood Flow Virtual Seminar Series for 2021. Pat Prasad and I are very happy to welcome you all today to our very first session for the year. It's an exciting session that we'll have today. It's a unique format that we have as a journal club. So I'm going to pass off in a moment to our chair, Baraa Al-Khazraji, who'll be chairing the session for us. But I just want to go over a few of the details regarding the series and some rules of engagement for you. So on the slide right now, I have the outline of the sessions that we have from now through till June of this year. So keep an eye out for those as the weeks progress. Also, just an update or a highlight of the Cerebral Autoregulation Research Network. If you'd like to be a member of this network, please email me your CV and I'll add you to our mailing list. In terms of rules of engagement for the session today, as always, please keep your microphone off and your video off as well. And then if you have questions at the end of the session, we will ask you to either post those questions in the chat or you can raise your hand and Baraa will ask you to turn your mic on and we can have you ask that question. As always, these sessions are recorded. They're being recorded right now, and then we'll post the recording on the CARNet website at the end of today or tomorrow when it's being processed. So thanks, as always, for your support of this series. I hope you enjoy the session today, and I'm now going to hand over to Baraa to introduce the speakers and just give a brief overview of the structure of the session. And we look forward to your engagement as the session continues. Thanks so much, Baraa.
Thanks, Caroline. So hi, everyone. Welcome to our first journal club as part of this ongoing Cerebral Blood Flow Seminar Series. Thank you so much to Patrice and Caroline for organizing nearly an entire year's worth of these online seminars. So my name is Baraa Al-Khazraji, and I'm a faculty member in Department of Kinesiology at McMaster University, and I'll be the chair for this session. So if you have been tuning in with us before, you'll know that we've traditionally had our seminars with a keynote speaker talk followed by shorter talks under a more common theme. Today's a bit of a different setup as it's a journal club focus where we invited authors of a recently published paper who will give a presentation on their work for 15 minutes, followed by a 30-minute Q&A between our trainee panel and the authors. We are very delighted to have Dr. Phil Ainslie and PhD candidate Hannah Caldwell present their manuscript titled Arterial Carbon Dioxide and Bicarbonate rather than pH Regulate Cerebral Blood Flow in the Setting of Acute Experimental Metabolic Alkalosis, which was recently published in the Journal of Physiology. Our trainee panel is made up of Audrey Drapeau, a PhD student in the Department of Kinesiology at Université Laval, Garen Anderson, a PhD student in the Department of Physiology and Anatomy at University of North Texas Health Science Center, and finally, Justin Sprick, a postdoctoral fellow in the Department of Medicine at Emory University. They have prepared a great set of thought-provoking questions, and we're very excited to have them have a discussion with our invited authors. So after the 30-minute trainee Q&A, we will have time to open up the floor to any questions. And throughout the session, you can feel free to either send me a direct chat and you can indicate to me if you'd like to keep it anonymous, or feel free to send it in the open chat and I'll read it out loud when that time comes. And you can also unmute yourself at the end. So without further ado, I'd like to invite Dr. Ainslie and Hannah to share their screens and commence their presentations.
Thank you, Baraa. Can you hear me okay?
Yes.
Thanks, Pat and Caroline, for organising this. I think everyone has been very complimentary. It's been a terrific thing to do for all of us stuck at home. Let me share my screen. Great. Okay, thanks for this chance to do this. I haven't done this journal club in this format before, but this is a study we've recently published. I just want to give you some context. It's led by Hannah, who will really do the business on what we did and why we did it. But Hannah's a real superstar. She finished her comprehensive exams just in December, and it's probably one of the strongest comprehensive exams I've ever seen in about 40 candidates. And she's really good. She cares about education, about science. She's careful in the lab with analysis, loves looking at old literature. Many times I send studies from the 50s and 60s to students I know, people I work with. And when I send them to Hannah, she's already read them. It really does blow me away. This study happened with the help of Jatinda Minhas from the UK. Jatinda came over a couple of years ago on a visiting fellowship for a month. And him and I have a strong interest in acid-base balance. And this is one of the first studies we sort of formulated to run out of a bigger programmed to do with acid-base balance and brain blood flow. And Jutena is great to work with from a physiology and a clinical perspective. And we managed to persuade Hannah to take this on and lead it. And of course, without Hannah, it never would have happened. But it's an emerging theme for us about acid-base physiology. So I just want to give a bit of a primer on why we're interested in it and why we think it's important, and lead into Hannah's paper. Okay, so this is a study we did in our lab about 11 years ago. I still remember it because I was in the lab testing and controlling arterial blood gases. But we measured arterial blood from the radial artery. This is arterial PCO2. This is a change in blood flow here. What we did here in about 16 or 20 healthy volunteers, we wanted to see what the tolerance was in the human brain to changes in CO2. So we clamped CO2 with a RespRack system from Joe Fisher, and we did it in 10 millimeter steps and then 5 millimeter steps. And it's interesting when you look at this, only 3 out of the 16 volunteers got to about 65 millimeters mercury above resting before really pulling off the mouthpiece. And that's a real tough challenge if you're clamping arterial PCO2. When you look at what's going on there as well, blood pressure is higher, cranial pressure is higher, sympathetics through the roof. It really is a huge stress. But it's interesting, I'll talk a little bit about that later, that there's many patients with chronic lung disease walking around with a resting PCO2 at 60, but they're otherwise feeling okay, despite the acidotic stimulus. And that's because it's been corrected from an acid-base perspective and bicarbonate. And I'll talk about that. Now, when we get people to hyperventilate, when we blow off CO2, you can see how, only half the group got below to about 15 millimeters mercury for more than 5 or 10 minutes. And you're very ischemic here. I mean, you drop flow by 50% to the brain. And there's only so much the brain can do about that. So we don't cope very well with extremes of CO2. It's interesting that the slope is a little bit steeper in the hypercapnic range, a little bit blunted in the hypocapnic range. And Hannah will talk a little bit about that as well. What we found in the study as well was the initial evidence of dilation of the extracranial arteries during these extremes of CO2. And, directly or indirectly, this led on to this slew of MR studies, which showed also that, some of the small arteries like the MCA are vasoactive with changes in CO2 as well. So interesting study, and we enjoyed doing it at the time. But, there's nothing too new about CO2 in the brain. We've known about that for well over a century. Probably the best study, or the first study to quantify it with, invasive techniques and in it washover approaches, was by Kenneth Smith in the late 40s. And what they've done here is they have a resting arterial pH. They've got cell blood flow here, absolute cell blood flow here because they've measured it. And they've induced some acidosis. This is respiratory acidosis by breathing CO2. Typically, respiratory acidosis is defined as a drop in arterial pH below 7.35. And you just see how in half a dozen volunteers, they become very acidotic and flow is going through the roof. Now, they've also got the volunteers to hyperventilate, and this causes alkalosis. Alkalosis is typically defined as a pH over 7.45. So you see the shape of the curve has been known for a long time. They use CO2 to change pH, of course, and this really highlights the important point of this seminar, that CO2 really is the effector which crosses the blood-brain barrier and then affects pH. It's not necessarily arterial pH which is the stimulus, it's a perivascular pH, but the pH is probably a closer metric of what's happening in terms of vasorelaxation in the brain. So this is one of the probably the most common fundamental equations in biology. I'm sorry that most people have seen this. It's a Henson-Hassleback equation, and just tells you that pH is determined by 6.1, which is the dissociation constant of carbonic acid, times by the log of plasma bicarbonate, divided by the arterial PCO2 times by the solubility in the blood. And the way I typically think about this or teach this is that really pH is determined by two things at a fundamental level. It's determined by the lungs, which affects arterial CO2. And we know from the alveolar gas equation that even small changes in alveolar ventilation here will have a profound effect on arterial PCO2 and therefore pH. So you can imagine if you double alveolar ventilation here, you half CO2, and you really and you cause a marked change in pH. Likewise, if you double alveolar ventilation, then you can affect this as well, of course. The other side is the kidneys. So if the kidneys retain or excrete bicarbonate, it can affect pH. So this is interesting, right? Because if pH is affecting tone in the cerebral vessels, which is, of course, then changes in can, but there's primary and secondary compensation. So if CO2 more, even for more than 30 minutes or so, if that change in CO2 bicarbonate. So for someone with serious lung disease who might have a 60s or 70s, typically these patients will also present with a very, very high bicarbonate, and pH will be either, partially corrected back towards normal, probably not fully corrected, but certainly move back towards normal. And we mentioned in Ryan's sort of review, and Ryan is another superstar in this field, and he's superseding myself and everyone else, which is great to see. But in Ryan's review, we mentioned a small section about buffering and blood-brain barrier, but not a lot, because it's quite a complicated topic. But do note there's evidence, preclinical evidence, that bicarbonate has direct effect on cerebrovascular tone, so can CO2, but we typically think that perivascular pH is the main mechanism. What we didn't have a chance to write about in Ryan's review was really about the interfaces between the blood and the brain. So remember, when we sample pH from the artery, we're really measuring it in the plasma. And the brain cells are seeing a different kind of pH. This is also influenced by the extracellular fluid, which is influenced by the CSF, and transport across the brain barrier. So it's very complicated, which is a good thing for us in science. But it's worth keeping in mind that when we change acid-base balance, there's also some other buffering and acid-base systems locally in the brain as well. So I just want to point this out just to think about different mechanisms which regulate cerebrovascular tone in humans. So of course, who cares? I always put this up here, one of my favorite cartoons, but I'm not going to go through these, but there's well over 100 acid-base disorders. So whether you're thinking about the effects of opioids or sleep apnea or, some carbonic anhydrase inhibition, some alkalosis, there's various forms of acid-base disorder. And unlike other aspects of brain blood flow, we hardly know anything about how these acid-base derangements affect brain blood flow. And that really fascinates me, because there's a myriad of mechanisms by which these different disorders can affect the brain, probably in an adverse way. So this is our first study. We've done a few of these studies to acid-base regulation, but this is something we want to carry on a program for the next 3 to 5 years. I find it fascinating. It's great people like Hannah and Jatinder and other collaborators who are interested in this work as well. So without further ado, I'll pass over to Hannah, who will expand on my few slides.
Perfect. Awesome. Well, thank you everyone so much. And I'd like to extend a great thank you to Caroline and Patrice for organizing this amazing seminar series, and to our chair, Baraa, for the kind invitation to present today. So I'd first like to just describe a few key proposed pathways contributing to acid-base regulation in the context of the current study, just building on a little bit of what Phil was just discussing. So firstly, we know that any systemic change in intravascular hydrogen ion and bicarbonate can indirectly affect perivascular or interstitial fluid pH, and that's through the interconversion to carbonic acid and production of water and carbon dioxide, which will diffuse across the blood-brain barrier. Additionally, according to the Henderson-Hassleback equation that Phil just described, intravenous sodium bicarbonate infusion will elevate arterial bicarbonate concentration and therefore arterial pH. And this would theoretically increase buffering capacity, so such that we would see that a given change in arterial carbon dioxide will result in a smaller change in arterial pH. Additionally, increases in arterial carbon dioxide will facilitate passive diffusion of CO2 across the blood-brain barrier. And this is going to lead to increases in intra and extracellular hydrogen ion concentration. And this is in accordance with the Chatelier's principle, so via shifting of the reaction equilibrium through continuous conversion of CO2. Additionally, increases in arterial carbon dioxide will also elicit an additive shear stress-mediated vascular smooth muscle cell vasodilation. And this is via increases in intracellular calcium, as well as AKT signaling to upregulate endothelial nitric oxide synthase activity and cause nitric oxide-mediated relaxation. Additionally, we know that some evidence has suggested that acute sodium bicarbonate infusion will reportedly elicit intracellular acidosis, or in other words, a reduction in intracellular pH. And this is via an exogenous bicarbonate-mediated rapid diffusion of CO2 and consequent increases in hydrogen ion concentration. And this has been theorized to contribute to the observed elevations in resting cerebral blood flow following a sodium bicarbonate infusion. And lastly, and we'll talk about this a little bit later, but there's been some recent preclinical work demonstrating a direct effect of circulating bicarbonate on vascular smooth muscle cell tone, such that alterations in extracellular bicarbonate concentration can directly affect vascular smooth muscle cell relaxation and contraction via alterations in intracellular calcium sensitivity. So these are just a few of the key pathways that perhaps we'll discuss later as they relate to the current results of this study. So the hypotheses for this study were that with elevated arterial pH following sodium bicarbonate, we anticipated a higher resting cerebral blood flow during poikilocapnic or uncontrolled room air breathing, and this would be due to relative respiratory suppression. So just in other words, as we induced an acute metabolic alkalosis, we anticipated some degree of partial respiratory compensation. Additionally, if we look at this cerebral blood flow versus arterial carbon dioxide relationship, or our typical CO2 reactivity response, we anticipated a downward shift in this relationship, such that we expected a lower cerebral blood flow for any given increase or decrease in arterial carbon dioxide versus the pre-sodium bicarbonate trials. And lastly, we also anticipated a blunting of this response. So in other words, a blunted CO2 reactivity due to improved arterial buffering capacity following the sodium bicarbonate infusion. So we had 11 healthy young males complete this study. And firstly, participants were instrumented with a radial artery catheter for repeated arterial blood gas sampling. We then completed baseline measurements, including cerebral blood flow and cardiorespiratory metrics. And then we completed a stepwise standardized CO2 reactivity protocol. And for this, we utilized a custom designed dynamic and tidal prospective target forcing system, where we first elicited 2 stepwise stages of hypocapnia, so -5 and -10 millimeters mercury arterial carbon dioxide. We then gave a 10-minute washout or rest, followed by 2 stepwise hypercapnia trials, so +5 and +10 millimeters of mercury arterial CO2. Each of these CO2 reactivity steps lasted approximately 5 minutes, we had a 5-minute steady state wash-in prior to the arterial blood gas sampling and cerebral blood flow measurements. We then gave a 30-minute intravenous sodium bicarbonate infusion, and this was calculated on an individual basis according to body weight. We then completed a poikilocapnic baseline, so again, just uncontrolled room air breathing, followed by an isocapnic baseline, where we clamped each individual's resting arterial carbon dioxide and oxygen values to their pre-sodium bicarbonate baseline values. And then we completed the same matched stepwise CO2 reactivity protocol prior to sodium bicarbonate. And at each stage, we measured global cerebral blood flow as the bilateral sum of the right internal carotid and left vertebral artery. For that, we used carotid duplex ultrasound, whereby we simultaneously measured arterial diameter and beat-by-beat pulse wave blood velocity to calculate blood flow in each vessel. So now to discuss our results. Firstly, we'll look at the resting cerebral blood flow. So we have on the y-axis here the absolute cerebral blood flow, and here we're looking at the poikilocapnic, so uncontrolled room air breathing. We have pre-values in blue and post-values in red, looking at the individual data, and we can see that post-sodium bicarbonate, during poikilocapnic breathing, we saw approximately 17% increase in resting cerebral blood flow. Next, if we look at the Isocapnic trial, so again, pre versus post sodium bicarbonate, we saw a 7% increase in cerebral blood flow, even though arterial carbon dioxide was clamped to the pre-resting value. And we can look at this further with the individual data. So here we have in the downward triangles, the absolute arterial carbon dioxide change value at rest And so we can see in the Isocapnic Trial, approximately 1 millimeter mercury increase in arterial carbon dioxide corresponded with a relative change of 17% cerebral blood flow in the upward triangles. And if we look at the Isocapnic Trial, we can see when arterial carbon dioxide was clamped or not different pre versus post, we still saw a 7% increase in cerebral blood flow. Next, if we look at some of our arterial blood gas values, in the table here, we can see a few key variables pre versus post sodium bicarbonate. And firstly, we can see that essentially the intervention was effective and that we were able to induce an acute metabolic alkalosis. So post sodium bicarbonate, we see an increase in arterial pH as well as bicarbonate, whereas by design, arterial carbon dioxide was clamped or not different pre versus post. If we look at figure A, here we can see the relationship between arterial carbon dioxide on the y-axis and hydrogen ion concentration on the x-axis. Again, we have pre-values in blue and post values in red. And this is looking at the average values at each stepwise stage of CO2 reactivity. And we can see that post-sodium bicarbonate, there was a leftward shift in this relationship. So that indicates that at each match stage of arterial carbon dioxide, we saw a reduction in hydrogen ion concentration, or in other words, an elevated pH following sodium bicarbonate. Likewise, if we look at the relationship between the absolute cerebral blood flow on the y-axis and arterial hydrogen ion concentration on the x, we see the very same relationship in that post-sodium bicarbonate, we have a leftward shift in this response, indicating that at each match stage of CO2 reactivity, we saw the same absolute cerebral blood flow response, irrespective of a lower hydrogen ion concentration, or in other words, a higher arterial pH. And importantly, if we look at this relationship between absolute cerebral blood flow on the y-axis, now indexed against arterial carbon dioxide on the x-axis, we can see that pre versus post sodium bicarbonate, this response was exactly the same. So in other words, at each match stage of CO2 reactivity, we saw the same absolute as well as relative cerebral blood flow response. Importantly, if we look at these relationships as cerebrovascular conductance, so accounting for changes in arterial blood pressure, we see the same relationship. So here we have cerebral blood flow, cerebrovascular conductance on the y-axis, and arterial hydrogen ion concentration on the x. You can see again that post-sodium bicarbonate, we have a leftward shift in this response. And again, when we index this against arterial carbon dioxide, there was no change pre to post in this response when we look at cerebral blood flow, cerebrovascular conductance. Next, we'll just briefly look at some of the acid-base buffering results. So again, just as a reminder, an improved buffering capacity would indicate a smaller change in arterial hydrogen ion or pH for a given change in arterial carbon dioxide. And that's just based off of the Henderson-Hassleback equation presented here. And so firstly, if we look at the absolute change in pH per absolute change in arterial carbon dioxide, again, we're looking at pre versus post sodium bicarbonate. We can see in the hypocapnic range, there was no difference in this absolute change in pH response. However, if we look at the hypercapnic range, we can see that post-sodium bicarbonate, we saw a larger absolute reduction in arterial pH, which would indicate a reduced buffering response post-sodium bicarbonate, at least in the hypercapnic range. Next, if we look at the absolute change in arterial hydrogen ion concentration per absolute change in arterial carbon dioxide, we can see in the hypocapnic range that there was a smaller absolute change in arterial hydrogen ion concentration post-sodium bicarbonate, perhaps indicating better buffering in this range. However, again, when we look at the hypercapnic values, we see a larger absolute change in arterial hydrogen ion concentration per arterial CO2. So again, indicating reduced buffering in the hypercapnic range. So lastly, if we look at our CO2 reactivity results, firstly, we'll look at the relative change in cerebral blood flow per absolute change in arterial hydrogen ion concentration. And we can see that post-sodium bicarbonate, there was an increase in hypocapnic CO2 reactivity. So just to remind you, on the previous slide, we just discussed that there was a smaller absolute change in arterial hydrogen ion concentration during hypocapnia. The relative cerebral blood flow response was the same, and therefore this explains the increased CO2 reactivity. Additionally, if we look at the hypercapnic CO2 reactivity, we see that post-sodium bicarbonate, there was a reduction in this hypercapnic CO2 reactivity response. And again, to remind you on the previous slide, we saw that the absolute change in arterial hydrogen ion concentration was larger. The relative cerebral blood flow response was the same, and therefore that explains this reduced CO2 reactivity response in the hypercapnic range. Lastly, and importantly, if we look at the CO2 reactivity response indexed against arterial carbon dioxide, we saw no difference pre versus post sodium bicarbonate in either the hypocapnic or the hypercapnic trial. So these changes in CO2 reactivity were only apparent when indexed against arterial hydrogen ion. Great, so in summary, we've seen that total cerebral blood flow was unchanged irrespective of a higher arterial pH at each matched stage of arterial carbon dioxide, indicating that cerebral blood flow is acutely regulated by arterial CO2 rather than arterial pH per se. Additionally, we saw that the cerebrovascular responses to changes in arterial hydrogen ion and pH were altered in keeping with the altered relationship between arterial CO2 and hydrogen pH following sodium bicarbonate. So in other words, the changes in arterial buffering capacity explained the observed changes in CO2 reactivity. And lastly, total cerebral blood flow was approximately 7% higher following sodium bicarbonate during isocapnic breathing, providing an additional evidence for direct vasodilatory influence of bicarbonate independent of arterial carbon dioxide levels. Excellent. So I'd just like to thank my team, particularly my supervisor, Phil Ainslie, all my project teammates and lab mates, as well as our collaborators in the UK, Jatinder Menhas, Ronnie Panerai, and Thompson Robinson.
Awesome. Thank you so much, Hannah and Phil, for that. So right now, I'd like to pass it on to Audrey, Garen, and Justin to carry on with the journal club discussion.
Thank you so much. First of all, just want to congratulate you both on that study. It's a difficult study to do, and it's really mechanistic for human work. So I'm excited to get more into it. To just kind of start off, we had a question as we were going over this article about the use of the end tidal forcing system for clamping CO2. Maybe if you could just point out exactly when it was on subjects, and you do mention a little bit in the discussion about the added dead space that system creates. Could you just comment on when it was used and how that might be affecting some of the results you guys were seeing?
Absolutely, that's a great question, thank you. So just to clarify, we were using the end tidal air force system essentially for all measurements apart from the poikilocapnic baseline values. However, for all of our respiratory measurements, we had the participants on the same mouthpiece setup, so the same mouthpiece pneumotac filter system. Just during the poikilocapnic breathing, we weren't controlling the gases that they were breathing. They were just breathing freely from the room. With respect to the dead space volume, so I mentioned that in the discussion, I just wanted to kind of clarify why we didn't see the anticipated increase in arterial carbon dioxide following sodium bicarbonate. So there's a couple of studies that show that when you give sodium bicarb and you elicit this acute metabolic alkalosis, you'll get some partial respiratory compensation. So participants, we expected were going to hypoventilate a little bit and cause an increase in resting CO2. After reading a little bit better detail about these studies, I realized that some of the studies that reported that 4 or 5 millimeter mercury increase in CO2, they actually did their respiratory measurements in a subset of participants, arterial blood gas in another subset, and then some alveolar ventilation values. So they were all taken from different subjects. And they had kind of explained as well that when you ask a participant to breathe on a mouthpiece, you might see there'll be an increase in physiological dead space that's going to affect your resting end tidal values, but also participants may actually just involuntarily hyperventilate a little bit on a mouthpiece, which would maybe mask some of the respiratory compensation we were hoping for. So that was kind of, I guess, the rationale, at least that we think, why we maybe didn't see that increase in CO2 post-sodium bicarb. Does that kind of help answer your question?
Yeah, no, thank you. I appreciate that. So, and then just kind of a follow-up. So were they, was end-tidal CO2 clamped then for the whole protocol, other than just the poikilocapnic, so during the infusion period as well?
No, that's a great question. So during the IV infusion, participants were just relaxing on the bed. We weren't taking any measurements during that time. The end-tidal airflow system was used for all of the CO2 reactivity. So at the start of the day, we had participants, the baseline measurements were just done. We collected room air values, and then whatever the room air sort of steady state CO2 and O2 values, we then clamped those throughout the isocapnic baselines, as well as the stepwise changes in the CO2 reactivity protocol.
Okay. No, thank you. That makes sense. And so this next question is kind of for both you and Dr. Ainslie. Prior work that you've published, Dr. Ainslie, has done cerebrovascular reactivity during high altitude ascent, which takes not sodium bicarb, but arterial bicarbonate concentrations actually in the opposite direction of this study. So you're reducing those with high ascent exposure. So I was wondering if you could maybe talk about both sides of that equation. Now you have acid-base balance where bicarb is being lowered, and then you also have in this study where it's being increased, and maybe kind of compare and contrast the results from the two studies.
Yeah, sure. I'll maybe have a go, and then Hannah can correct anything I get wrong. But typically when we've got high altitude, we're in a state of, as you know, respiratory alkalosis, and then you get some metabolic compensation, which is really dumping bicarbonates. When you think about that Henson-Hassleback equation, certainly where we do research, bicarbonate's down to 15 and 16 milli equivalents. So when you do the same step change in CO2, and this is what Hannah talked about in her direction, when you do the same step change in CO2, you get a much greater change per point unit change in pH. So the stimulus is much bigger when you do it at high altitude because of the reduction in buffering. Hannah increased buffering, and at high altitude it's reduced, which is the same with pregnancy. It's the same with certain phases of the menstrual cycle. And high altitude is a great example, and it's a good example because it's a chronic change in acid-base balance with metabolic compensation. And it's amazing because when we first started doing these sort of step clamps at high altitude, we thought, well, we'll increase it per 5 millimeter step at sea level, then we do it at high altitude. And only about 3 or 4 people could get to +10 at high altitude rather than +15. We thought, oh, fuck, why is that? And then we realized that, of course, when we looked at the pH, there was a much bigger pH stimulus per given change in CO2 because of reduction in buffering. And it's really interesting because, depending on how you measure activity, it's generally higher at altitude, but is it really? Because, it's a physiological construct of a greater pH stimulus. Mm-hmm. And that really got me excited about acid-base balance and thinking about the 101 other situations where acid-base disturbances happen. And for me, it's educational because it highlights about Hannah's study was very acute mid-ablation of buffering, whereas high altitude or pregnancy or a few days of in the luteal phase, it's more of a sustained stimulus and acid-base balance where you get more compensation from the metabolic perspective. Did I get that right, Hannah?
Yeah,
Yeah, I think so. The only thing I was going to add, and maybe Garen, you could stay tuned for this, but we have a study that it's just in review at the moment, but we looked at CO2 reactivity at high altitude, and we did this with and without giving oral diamox. So we induced a metabolic acidosis at high altitude, and then we also gave again a sodium bicarbonate infusion to restore pH. So really just kind of screwing around with acid-base balance and looking at the effect on CO2 reactivity at high altitude as well. And again, kind of showing that it is regulated at least in these situations by CO2 rather than pH at high altitude as well.
Thank you. I'll look forward to that. That sounds really interesting.
Regarding the pH, we are interested in asking you your thoughts about the fact that you induced an alkalosis, a metabolic alkalosis with the infusion of bicarbonate. And what do you think the effect would be on the, or do you think that the infusion would create a compensatory mechanism in response to the reduction, the increase in pH for the oxyhemoglobin dissociation curve with the oxygen? Do you think that it might have an influence on the increase in cerebral blood flow that was observed in your study? I don't know if I asked my question clearly enough.
I think I understand. Phil, do you want to go for this one?
Yeah, sure. It's a great question. I hadn't really thought of that before, Audrey, but you would get a leftward shift of the dissociation curve because of the alkalosis. Certainly, as I'm sure you're aware, the brain isn't very sensitive to changes in PO2. You have to drop it below probably 50 millimeters of mercury at times. But it's not to say, however, that if we have caused a big shift in the dissociation curve, then we've done a desaturation protocol on somebody. It's not to say it wouldn't be different then. So I think that's a great point, because this study was focused on CO2, but you're right, shifts of the dissociation curve would affect the relationship between arterial PO2 and perhaps hypoxic vasodilation.
And in the case where you guys were studying participants that were at rest without any cerebral metabolic activation, how would you expect the difference if you were testing the participants while they were doing exercise, for example?
So I think for that, sorry, to just clarify, you're asking if we had done the CO2 reactivity following sodium bicarbonate, but then during an exercise protocol?
Well, it's not reported in your study that you were doing it during exercise, but I'm wondering how this condition might affect your results.
Right, definitely. So I think that's a really interesting follow-up study that we'd like to do. So, for example, if we were doing a prolonged exercise protocol and we saw a reduction in pH or sort of a metabolic acidosis during exercise, depending on the intensity, of course, I would be really anxious to see if a sodium bicarbonate infusion to correct for that metabolic acidosis could alter CO2 reactivity during exercise. And then just to further add to your point about sort of the oxyhemoglobin dissociation curve, that's an interesting note. I think a few studies have shown that when you give a sodium bicarbonate infusion, they've actually reported reductions in hematocrit. And that's due to a bit of sodium facilitated diffusion of water through the intravascular compartments. So they attributed their increase in resting cerebral blood flow to actually a reduction in hematocrit as well. So not necessarily a direct effect of PO2, but maybe related via a reduction in hematocrit. We didn't see really any significant changes in hematocrit in our study, so we don't know whether that was actually contributing to the cerebral blood flow results. But I think it's just kind of an interesting consideration that some studies have reported that with sodium bicarb.
Audrey, I think it'd be a neat question because when we get some hyperventilation at high intensity exercise and that helps return cerebral overflow back to normal or below normal. But I wonder about the addition of additional alkalotic stimuli on that response. Yeah, great idea.
I have a question regarding the potential mechanism through which bicarbonate may be causing vasodilation. So the reference literature describes an elegant series of experiments performed in isolated mouse cerebral arteries and suggests that reductions in bicarbonate may cause vasoconstriction through a pathway activated through receptor protein tyrosinase phosphatase gamma, or RPTP gamma. However, the effects were only present under reduced bicarbonate concentrations, so less than 22 millimolar. And there was no vasodilation in response to increasing concentrations. And in your studies, bicarbonate increased to approximately 27 millimoles per liter following your infusion, and you did see vasodilation. So I was just wondering how you reconcile these in vitro studies compared to your findings, or if you have any thoughts on that. Is this potentially just different species-related differences in this pathway, or do you think that maybe there's another yet to be identified sensor of bicarbonate that's responsible for the vasomotion and vasodilation you saw?
Yeah, that's an excellent question, Justin. Regards to the exact bicarbonate sensors in the brain, I think that's very new for us. I don't know that we have really any convincing evidence in humans or any in vivo evidence. We were kind of excited by these preclinical values with this sort of bicarbonate direct effect on the vascular smooth muscle tone. Of course, with the caveat that these are animal studies, we completely acknowledge that this is kind of preclinical work. It's likely not directly applicable to any of our human work, but we were just thinking that maybe if these results can be extrapolated to our current study, it may help explain a vasodilatory response to bicarbonate. And the reason that we had reasoned that is that because we saw that sort of 17% increase in cerebral blood flow during poikilocapnia, but then when we clamped CO2, so basically removed the effect of CO2, we still saw a 7% increase, and even I think there was a 10% increase in cerebrovascular conductance. So really the only explanation is that bicarbonate was higher, and maybe that's directly affecting the vasculature. But in terms of the exact mechanisms, I don't know if I can really speculate in humans at the moment.
Sure, exciting area for future work then.
Yeah, definitely.
Hannah, do you think it might be possible that there would have been an influence of the tonicity of the solution, maybe creating an intra-arterial osmotic shift? And do you think that this osmotic shift might have masked some of the effects of bicarbonate in the cerebral vasodilation? Sorry.
That's okay. That's a really great point. So we had considered that. So it was so the sodium bicarbonate that we gave was diluted with saline, and the total infused volume was 250 mils. So we think was such a small amount, and individuals were allowed to drink water ad libitum and go to the washroom as they wished. So we don't think we necessarily changed central blood volume or any intravascular volume with the infusion itself. But regarding the tonicity of the sodium bicarbonate, I haven't thought about that too much, but I don't know whether such a small amount of sodium bicarbonate would really affect whole body tonicity. I'm not sure. Phil, do you have any thoughts on that?
Sorry, I was just replying to a question from Jeff. I zoned
No
out.
problem.
Sorry, do you mind repeating it? I can help if I can.
Whether our sodium bicarb affected the tonicity of the blood, or because I think with the isotonic solution.
Yeah, it's a great question. I mean,
sorry, hypertonic solution.
Hypertonic, yes, sorry.
Yeah.
Yeah, it's a great question. I'm not sure of the answer. I know when we use the Henson Hassleback, it is quite a simplified. It's a simplified version of acid-base balance regulation, and it ignores all the strong iron differences between, blood and brain interfaces. So there could well be some changes in tonicity of the fluid. Yeah, that's a good point. Because if you do give a hyperosmotic infusion to someone, it can reduce intracranial pressure. It can do other things to the brain as well, independent of pH. Good point.
And I'm just curious because you were referring to Lambertson study where they were infusing bicarbonate, but they also used like you combined it with saline solution, and they combined it with a glucose solution. So I was wondering what made you choose saline solution rather than glucose solution?
Yeah, definitely. So a few different reasons. So we based our sodium bicarbonate and the hypertonic saline solution based on a study by Luc Tepema a few years ago looking at acid-base balance on respiratory control. So we were trying to sort of elicit the same change in arterial pH and bicarbonate. And then I guess the reason why we, so just to think about that Lambertson study, why they use glucose. I think another reason that we did the hypertonic solution with saline is there's been some studies looking at the effect of sodium bicarbonate in reducing intracranial pressure in patients with traumatic brain injury. And so a thought of this study was we were thinking of a clinical application to individuals with acute ischemic stroke. And so I think trying to follow sort of some of the same therapeutic guidelines that had been previously done in the patients with traumatic brain injury to see whether the hypertonic saline solution was going to affect acid-base balance.
I want to follow up with another clinical application in that regard. And one that I'd like to ask about is in regards to end-stage renal disease patients that are undergoing hemodialysis. Hemodialysis causes a reduction in cerebral blood flow, and this has been linked to cognitive dysfunction and over time, brain structural abnormalities as well. One goal of hemodialysis is correction of metabolic acidosis, and this is accomplished through infusion of supraphysiologic concentrations of bicarbonate in the dialysate bath, typically 32 to 37 millimoles, so pretty high. Based on the direct cerebrovascular dilatory effects of bicarbonate that you report, do you think that this influx of bicarbonate during hemodialysis may be one mechanism that is opposing reductions in cerebral blood flow during dialysis?
That's a really good question. Yeah, I hadn't really thought about like the direct effect of bicarbonate on cerebral blood flow in dialysis. I agree. I think that could be a nice explanation. Just trying to think what else would be contributing.
You certainly see quite large changes in viscosity and hematocrit, right? Just in during dialysis.
Yes, that changes as well. Mm-hmm.
Yeah, I think it's a great. In terms of populations with acid-base disorders, that the renal failure and dialysis is just a fascinating population. But yeah, I mean, there could well be some benefits to the bicarbonate. But it's certainly interesting because the kinetics of the acid-base changes with dialysis, my limited understanding, it's going to be very complex, right? Depending on dialysis and infusion and everything else, but fascinating from a cerebrovascular perspective.
Mm-hmm.
Sure. Are there any other clinical applications that come to mind? You mentioned stroke. We talked about dialysis. Are there any other applications that you'd like to see as some next steps in future directions in regards to this work?
Yeah, sure. I mean, we have interested in carbonic anhydrase inhibition from a metabolic acidotic perspective. We know a little bit of work in people who with severe COPD, but early stage. But those patients who are retaining CO2 in lung disease. We've done a number of studies now with people with polycythemia in South America, where we've hemodiluted them to look at how that affects brain blood flow, and it seems to go as we expect. But any acid-base disorder where there's respiratory metabolic changes. It's just fascinating from a human cerebrovascular perspective. It's probably, there will be more known preclinically, but the challenge is now to try and do it in humans and trying to optimize what is an ideal, acid-base status for cerebrovascular health.
Sure.
Maybe just one more quick question from us as far as study design or implications. So you have a sample of young, healthy men for this study, and you talk a little bit in the discussion of the article about how it might affect women. Do you have plans to do this in women and see how that comes out?
Yeah, definitely. That's a great question. Thank you, Garen. So that's exactly right. I think we want to repeat the study in women. And our intention to do that, rather than, for example, controlling for menstrual cycle to allow for inclusion in the current study, we felt that that would actually probably mask some of the sex-related effects of men and women, and rather that it would be more important to repeat the study in women. And particularly, I would be very interested to look at women in the luteal phase. And so in the luteal phase, with increases in progesterone, as kind of described in the article, actually acts as a respiratory stimulant. So women can at times be relatively hyperventilating and reduce with a reduction in arterial CO2, sometimes as much as 4 to 8 millimeters mercury at rest. Which also causes a reduction in base excess and buffering capacity. So I think it would be really exciting to look at whether a sodium bicarbonate infusion in women can actually help correct some of this respiratory alkalosis, and if that further affects their cerebral blood flow regulation.
Thank you. That would be really interesting to see. As far as kind of other populations that this kind of work has effects for or implications for, have you considered maybe aging populations, how this may change or these mechanisms might be different in aging? This is a big area of trying to understand what's changing over the lifespan with cerebrovascular health.
Exactly. And I think as well, something we'd kind of thought about in our clinical translation as well is if you look at the literature on CO2 reactivity and aging, it's a complete mess. Some studies say it goes up, some say it goes down, it stays the same. It's really hard to find consistent findings. And so we were actually wondering whether chronic changes in acid-base buffering with aging is actually contributing to some of the heterogeneity in these findings. And if that could help explain why we see such inconsistent results across studies. So I think that would be a really exciting follow-up, is to see how changes in acid-base balance in older individuals affects CO2 reactivity.
Sorry, Phil, I don't know if it was me that froze or I think you were saying something on the last comment.
I was saying quite some time ago, we looked at blood gases in dozens of people over the age of 70. In resting PCO2s and bicarbonates are markedly different in people over 70 because there's changes in structure and function of the lung and the kidneys. So it doesn't surprise me that there's huge variability in a cerebrovascular CO2 test in someone who's 70 versus 20 because of changes in buffering.
Okay, great. Thank you for that. So I see, Phil, you've answered one of the questions at least in the chat, but maybe we'll get you to expand on Jeffrey Dunn's question. So he asked, are you suggesting that CO2 is sensed in the lumen of the blood vessels, or could it be that serum pH doesn't directly relate to pH on the parenchymal side of the endothelial cells, while CO2 could diffuse to the other side? So the regulation of dilation could still be pH changes on the parenchymal side caused by CO2 dissociation. I'm wondering if you guys can comment on that.
I'm happy to do this. Hannah, I think you know this just as well. There's certainly, Hannah mentioned it in her paper and in Ryan's review as well, that there's direct in vitro evidence, preclinical evidence of a direct effect of a CO2 on the lumen side. and also some direct effects of the bicarbonate side. But I agree with Jeff fully, and this is what we try to raise, that the compartments, the kinetics of a change in plasma pH, for example, it needs to get to CSF, extracellular fluid, and there's strong ion differences there. There's different buffering systems in those different compartments. So I could never say that a change in pH equals 100% change in pH at the perivascular level, because it's much more complicated than that, of course, for those reasons. And that's what we don't know. We do have plans to do some CSF sampling soon, but it's still not as close as what we would want, but as close as we can probably get in humans.
Mm-hmm.
Hannah, do you have anything to add?
Yeah, I fully agree. Just rereading this question from Jeffrey, I think the latter half of his question is definitely 100% what we'd agree. I think it is really rather that it's the diffusion of changes in arterial CO2 across the blood-brain barrier that are going to affect perivascular or extracellular pH. That's going to be really important for alterations in cerebral blood flow. I do think that it is certainly the intrinsic capacity of the perivascular extracellular pH rather than arterial changes, but that is just to say that arterial pH per se or in and of itself isn't going to directly affect cerebral blood flow apart from the changes induced by CO2.
Okay, great. Thank you. Another question is, cerebrovascular reactivity is typically thought to be brought about by relaxation of smooth muscle cells located on the extravascular side. Does bicarbonate increase on the extravascular side, which changes in smooth muscle tone?
Cool, that's a great question. So, and we tried to discuss this a little bit as well with respect to the kinetics of bicarbonate across the blood-brain barrier. So typically, at least in this acute experiment, we don't actually think that our arterial changes in bicarbonate really did affect the perivascular extracellular pH or bicarbonate values in this study. Obviously, in a more chronic situation, we'd expect to see these changes in bicarbonate in the extravascular space. So I don't know whether that relates to the current study, but I think in a more chronic situation, we could expect to see bicarbonate directly affecting the extravascular space.
Okay, great. Thank you. And Ronnie Penray says first, thanks for the excellent presentations. And did you have a chance to observe any differences in cerebral blood flow time constants to either rising or decreasing values due to changes induced by either PaCO2 or bicarbonate?
Cool, that's an excellent question, Ronnie. We unfortunately didn't look at that in this study. For our CO2 reactivity stages, they were each a sort of 5-minute steady state wash-in, and then all of our measurements were kind of conducted in the steady state. So we didn't do any sort of dynamic changes in arterial CO2. We did, not reported in this study, but we did have TCD on as well, measuring middle cerebral artery and posterior cerebral artery blood velocity. So technically we do have that data in terms of any dynamic temporal changes in intracranial blood velocity, at least while we are making those changes in arterial CO2 prior to the steady state values. But we haven't looked at that yet. So maybe we can if you'd like.
Okay, awesome. And then our final question, at least that I see in the chat, is by Anthony Bain. First, he says, great work, Hannah. And he's curious, did you keep track of the ventilatory shifts with the bicarbonate infusion with the CO2 inhalation? So that is, how much less CO2 did the Air Force need to pump in for a given partial pressure of arterial carbon dioxide? My question is related to how much of that bicarbonate is shuttled to the cerebral spinal fluid, and are the transporter proteins across the blood CSF and blood-brain barrier altered with shifts in blood acidity?
Well, thanks, Tony. Those are really interesting questions. So to answer your first one regarding ventilatory shifts, so that is something that we were really interested in as well. whether the sodium bicarbonate was going to affect the respiratory response to our CO2 changes. And we didn't see any differences between ventilation, respiratory rate, tidal volume, anything like that during our CO2 reactivity, which we're maybe a little bit surprised by. However, as you know, with the Air Force system, but I'll maybe mention for those that don't, it operates independently of ventilation. So when we target a prospective end tidal oxygen or carbon dioxide value, it's going to adjust the fraction of inspired gas irrespective of how much the participant is breathing. So it wouldn't necessarily affect our cerebral blood flow results per se, but from a respiratory perspective, I'd have to ask my Chief Air Force operator if they noticed any changes at each CO2 reactivity. Just kind of anecdotally, whether we were giving more CO2, for example, for a given stage. I didn't notice that, at least, in the experiment. And then your final question here about bicarbonate transport shuttle to the CSF. That's a really good question. I don't know how transport proteins would be affected by changes in blood acidity. I'm not sure if Phil can elaborate on that, but I'm not sure for that one.
No, I'm not sure either. I know that the PCO2 is typically higher in the CSF, but the bicarbonate is somewhat comparable. But I'm not up to date on the nuances of the transport proteins.
Okay, thank you. So Caroline asked if you could expand a little bit more on your comments regarding differences in acid-base balance across different phases of the menstrual cycle.
Yeah, absolutely. So I can just describe again. So really with particular interest on the luteal phase, where we have an increase or an elevated progesterone levels in women, and that may act as a respiratory stimulant. So having females relatively hyperventilating in the luteal phase, and it's been reported that this respiratory stimulant can actually reduce arterial carbon dioxide by maybe as much as 4 to 8 millimeters of mercury. So you're hyperventilating a little bit, and that's also going to reduce base excess. So women could potentially be a little bit having respiratory alkalosis and affecting their buffering capacity just even at rest. So we'd like to see whether if you were to give women in the luteal phase who are experiencing this hyperventilation sodium bicarbonate infusion, if that can help correct for this reduced resting arterial CO2.
It's interesting. I love the idea of acid-base balance, because if the acid-base stimulus is metabolic in nature because of change in bicarbonate, then the compensation is respiratory, which happens really quick because, changes in alveolar ventilation have a very rapid control mechanism. But if the acid-base disturbance is respiratory in nature, i.e. in the luteal phase, for example, then the renal compensation happens slower, more so over, 6 hours, maybe a couple of days, depending on how much alkalotic stimulus there is. And it's interesting, the mechanism by which the respiratory alkalosis affects bicarbonate excretion is via PCO2 at the renal tubules. So it's really neat to think about the links between the respiratory and the renal systems and how they interact at different time domains to affect acid-base balance and then potentially brain blood flow, of course.
Awesome. So Caroline, thanks you both for that. And then Andrew Robertson asked, did you see any changes in waveform morphology that may be indicative of baseline tone changes following bicarbonate infusion?
Great. Okay, so Andrew, I assume you mean the blood velocity waveform. So I'm just thinking, yes, okay, great. So I can look again. I know kind of just anecdotally, I didn't notice anything in our when I was scanning the ICRVA. In terms of if we're looking at indicative of tone, we did see If you consider kind of the regional differences in internal carotid artery and vertebral artery flow, there was a small but significant increase in vertebral artery diameter post-sodium bicarbonate. So we saw sort of a time effect that at each stage of CO2 activity, the vertebral artery was slightly dilated. It caused a small increase in vertebral artery flow post-sodium bicarbonate, but the changes were so small that they didn't contribute to any meaningful change in global cerebral blood flow, which we were mostly interested in for this paper. Perhaps that could be indicative of a small bit of vasodilation, at least in the VA following sodium bicarb. But in terms of blood velocity waveform changes, I can't comment on that, unfortunately.
Awesome. Thank you for that, Hannah. I don't see any more questions in the chat. I don't know if the trainees have any residual pressing questions that you're hoping to get in. Feel free, Pat and Caroline as well. And like I said, anyone, feel free, you can unmute yourself if it's easier to speak rather than typing it in the chat.
Yeah, I'll ask one more while someone might be getting their final one typed there. It's regards to how you control for blood pressure. So as you know, there's kind of been a debate in the field of how we do this when we're doing cerebrovascular reactivity. One approach is to normalize it to conductance, or express conductance as a way to account for it. Another one is to include blood pressure as a covariate in your analysis. And it seems that you've done both approaches. You've normalized it, or you've accounted for conductance and you showed no change, and you also included it as a covariate. Just curious your thoughts on the rationale for that, why doing both, and if there's any specific reason that you chose to do it that way.
Yeah, absolutely. That's a great question, Justin. Yeah, I did choose to do both. So in our lab, we've kind of adopted the approach to do the linear mixed modeling covariate adjusted blood pressure. The reason that we kind of favor that approach is with the linear mixed modeling, it will account for any individual changes in blood pressure and to see whether that alteration in blood pressure is actually contributing to an effect on cerebral blood flow. A bit of the criticism for doing just the cerebrovascular conductance approach, so when you just take cerebral blood flow divided by mean arterial pressure, is there's a few assumptions associated with that. So firstly, you're assuming that the cerebral blood flow to blood pressure response is linear, which already kind of contradicts the kind of very premise of autoregulation. So that's a bit of an important assumption that I think we maybe sometimes forget about. And then kind of a caveat with that as well is that when you report values as conductance, you're almost assuming that any change was fully accounted for by an alteration in blood pressure. Whereas again, it could be a variety of factors affecting tone. So I guess the reason to present both, I favor the side of the linear mixed modeling analysis. I think that's kind of the stronger statistical approach. But just, I guess in the practice of reporting both for transparency and maybe especially to make a comparison between studies that have only reported conductance, that's a little bit easier to make a direct comparison between studies.
Thank you.
No problem.
It's a great question, Justin and Hannah, and applies to other studies too, beyond just looking at the question you did. So that's great. Binu Thomas, thanks you for answering the previous question, and again for a very nice presentation. And they're wondering, is there any information on the time it takes for bicarbonate to cause vasodilation and cerebrovascular reactivity changes?
Cool. So that's a great question. So as far as I'm aware, if you were to do an intravenous sodium bicarbonate infusion into sort of systemic circulation, it will actually take several hours for that change to be seen in the extravascular fluid or the interstitial fluid. So I think it would be sort of several hours before we'd actually see a central change in bicarbonate or pH. There's been a really nice study I think it was in the 50s or 60s, in anesthetized cats, where they gave a sodium bicarbonate infusion and then measured extracellular pH and bicarbonate, and showed that extracellular pH increased very markedly. However, it was restored within the first hour, irrespective of a persistent elevated pH in the systemic circulation. So there was sort of this initial rapid increase in extracellular pH, but it was rapidly restored within an hour. So it's really difficult to say. At least in our study, I don't believe we really affected extravascular pH, at least acutely. So I think it would probably take several hours to see a change directly by the bicarbonate in the interstitial fluid.
Out of curiosity, how long does your study protocol last? Like one session?
Absolutely. So for this study, I think we were averaging about 7 hours per person, but I can explain why. So in the study we had, we did CO2 reactivity, the brain blood flow scans, and then we also did a 5-minute neurovascular coupling test at each stage of CO2 reactivity. So in total, each stage itself lasted probably 15 or 20 minutes, and then we move on to the next one. So in total, I guess, yeah, like including, it takes about an hour to do the radial artery catheterization in the beginning, and then we have setup, and then do the first trial, and then the 30-45 minutes for the wash-in. I think we were averaging about 7 hours per person. Yeah.
You were talking about the study about the anesthetized cats.
Yeah.
How you were saying that the pH like kind of balanced out after a couple of hours. Do you think it might have affected your study if it lasted 7 hours? Like, do you think that the CSF might have, I don't know, washed up or rebalanced the pH?
Yeah, that's a really good question. So I guess it is probably something that we don't know. So depending on like how long it took per individual, like we did try to start immediately after the bicarbonate infusion, and then each stage was conducted in the same order for each individual. I don't know, even still, like, so it would have been, I guess, the second half, so the post-sodium bicarbonate measures probably only took an hour or so. It's really a lot of kind of time at the beginning with setup and experimentation and everything that takes the majority of the testing session. So I think our post-sodium bicarb CO2 reactivity was only maybe the last hour or so. So I don't think it would have affected our results. It's a great question, though.
Okay, great, thank you, Hannah. I don't see any more questions in the chat. So I guess I'll pass it on to Pat to maybe share with us information for our next session. And thank you so much, Hannah, Phil, Garen, Justin, Audrey. This was a really cool interaction. It's kind of like everyone's having a lab meeting at the same time. It's really great. So thanks so much for that.
Thank you.
Yes, thank you very much. And I would also like to take this opportunity to thank the contributors. I cannot speak for Carolyn, but it was totally what we expected with this journal club. So it will definitely set the bar high for the upcoming journal club style discussion. And I'll just, okay, here. So make sure to register for the next more traditional Cerebral seminar series. It will be also an amazing seminar. So it will be chaired by Dr. Jenna Kenner-Storver, and it will be about near-infrared optical methods for assessing autoregulation, intracranial pressure, and neurovascular coupling changes. So again, thank you for your attendance, and we are looking forward to the next to seeing you in 2 weeks. Thank you to everybody.
Yeah, thank you, everyone. And I absolutely agree with Pat. That was an excellent session. So thank you to Hannah and Phil for agreeing to present today, and to our panelists, Audrey, Garen, and Justin. You did an outstanding job. And thank you also to Baraa for great chairing of the session. So Thanks, everyone. We hope to see you soon.